Okay. So this class is Protein Database, Protein Visualization, and Structural Bioinformatics, MBSB 501. I am Dr. Duangrudee Tanramluk at Institute of Molecular Bioscience, Mahidol University. You can access my website at ajanvi.wordpress.com. So the website that have my lecture and also everything is this one in addition to the Google class. So I will have many videos about my drug design platform and also the courses I taught, including basic bioinformatics, structural bioinformatics, and protein visualization, which I will cover in this class. So the assignment would be on this Google class, which have the interactive slides, and also I'll have assignment. So the class is Protein Database Visualization and Structural Bioinformatics. The reference to this class would be Introduction to Bioinformatics by Arthur Lesk. So I will briefly introduce about biological database and familiarize you with databases and visualization tools used for protein structure analysis. I want to give you hands-on on practical problem on data retrieval using structural database query forms. So what is the database? Database is a kind of electronic filing cabinet. It's a repository container for a collection of computerized data. So the characteristic of structural database, including the content and the ontology, interrelationships among the data, and format of data. So the content would be basically the protein data bank formats, and the ontology is the set of predefined terms that used to describe the interrelationships among the data based on the characteristic or the function. And the format of data is the .pdb or the protein data bank file format. And the rule of selective retrieval would be various depending on which website you access. So it also provides links to other data sources. Normally the web-based version of UniProt or other databases will allow you to link among various data sources. So the type of databases, according to the content, that is the primary data source that are the originally created version of the data. Basically, the protein data bank file would be the one that protein crystallographers deposit by themselves into the protein data bank, mainly the RCSB protein data bank and protein data bank in Europe, for example. So these primary databases are responsible for data curation, and they try to maintain the same versions across several databases. And there is also the derived database, which take the data from primary databases to recombine, re-annotate, reformat, or re-represent. So the example I will give for this database class would be the CAS database, which have the class architecture that are derived from the structural database into the fold of the protein. So they gather it and re-annotate it into the class of the protein according to the shape. And then they deposit it with a different IDs in order for you to withdraw the data. And they provide different environments according to the type of the database that they offer. So why are we studying structural database? Given a protein sequence, a structure, or the ligand, you could be able to find a structure that corresponds to these three characters and should allow fast and flexible query and integrity maintenance across several databases. And it should also enable you to data mine, do the machine learning or something to process the data. So the structural bioinformatics data, the protein is normally, it is called a protein data bank because it's normally the main class of the structure would be the protein. It will link for the smallest ones such as trypsin inhibitor, which are 36 angstrom across and have molecular weight 8 kilodalton. And this is botulinum toxin, which is 1,300 residues. So this one used for Botox, as you can see that it makes your face look nicer. So this is the structure of botulinum toxin. And it may be as large as 50S ribosomal subunit, which comprises 2,833 nucleotides. So it will be large. And lately, with the invention of cryo-EM, the structure is extremely large, like the whole virus or even larger. So the structure of the protein, these ones have several shape and size and characteristics. So for example, let me pick the highlighter. This one, laser pointer. This one is the GROW-EL structure. The GROW-EL is like a barrel shape. It has a top-like characteristic, so it's like tongue. So there is the small protein that gets into the GROW-EL structure, and then it closes the lid, and then it shakes, and then the protein inside would be nurtured and folded correctly in the proper form. So these proteins have the shape that allows it to perform certain function. So there is structure such as this one, rhinovirus. So it's globular shape. This has the Y shape of antibody. And these are the protein in electron transport chain, like ATP synthase, or these ones is the structural proteins such as microtubules or actin filaments or collagens. So these ones, all these proteins have a 4-letter identification number leading to the structure of them. So you can use this 4-letter in order to retrieve the protein structure data from the protein data bank. So the primary structure of the protein, normally we have the sequence, and the sequence, once they fall into the repeating torsions, like this one in spiral shape, we will call it alpha helix secondary structure. Or if it go into the right and going to the left like this, we call it beta pleated sheet. So this is alpha helix beta sheets. And if it has a mixture of alpha helices and beta sheet together into sets like this, alpha, alpha, or beta, alpha, beta. This is called the motif. If the motif come into a recognizable fold that we can relate to the daily life, like basket, barrel, prism, solenoids, or clams or sandwich, they will be called the domain. And if it has a secondary function, if it has the ability to perform its function into the mixture of domain, or sometimes the domain itself have functions, it will be called tertiary structure. And if it has cooperativity among each unit, it will be called a quaternary structure, as we can see in hemoglobin in various subunits. So we have protein sequence that can adopt 3-dimensional structure with various conformation and dynamics. For basic building block of protein, we have this amide group, have the R group that are specific for each amino acids, and have the carboxylic acid. So these ones, once they become the polypeptide chain, they will be called amino acid sequence. So the R group will be varied. And normally in nature, the amino acids have the L left-handed chirality, which means it has the left-handed character going from the largest molecular weight to the smallest one. So it goes from CO to R to N. So this is a familiar picture of periodic tables. In protein structure databases and structural data, the row that we use often is this row. So you only need to remember 4 colors. So it's this row. The carbon in white, nitrogen in blue, oxygen in red, and sulfur in yellow. So these amino acids, when they become the structural side chains, you can see they have various shapes and size. The smallest one is glycine. So glycine, you only need to understand that it has the smallest structure, and it allows for flexibility of the main chain when it resides. So normally glycine allows for flexibility, and alanine, valine, leucine, and isoleucine. This one doesn't have colors, so they are hydrophobic, small or medium size. They normally serve the structural roles. So it's not very interesting, but they serve the structural role with its various size and shape. And serine and threonine have one hydroxyl group, which is the oxygen gamma here, oxygen gamma. So the red tips is OH or hydroxyl group. It's allowed for phosphorylation or other post-translational modification. Cysteine allow for reducing agent as in cysteine bond formation. So there is another cysteine come besides. And when you want to modify the protein in order to increase the thermostability, you engineer so that it has 2 cysteine binds together and the protein becomes stronger and resist to heat, for example. So this cysteine used a lot in industry in order to modify the protein to have more stability. For aspartate and glutamate, they have double bond OH. So in electron density, they look the same, but it's basically this one is double bond OOH. And if it has 2 red tips, if it has 2 reds, it's a negative charge. So it's partial negative charge. The partial negative charge allows it to form salt bridge with partial positive charge. So if it's red, it forms salt bridge with the blue colors. For asparagine and glutamine, this has the amide bond. So it has the amide group, not bond, amide group. So it has a double bond O and NH2, for example. So it has polar uncharged characters. So it has 1 red, 1 blue. So this one polar uncharged character. Asparagine and glutamine is different by 1 carbon. So glutamine is 1 carbon longer than asparagine. And glutamate is 1 carbon longer than aspartate. So the end of the side chain or the penultimate group is the same, but it different by 1 carbon size. So this 1 carbon is longer, this 1 carbon is longer. And in the real structure, this has rotatable bonds. It rotates around and it has several torsions angle. So this is just 1 rotamer that you can see, but in the real structure, there would be several conformations. For the lysine, arginine, and histidine, these ones has partial positive charge characters. you can see in blue. So if you see the colors, you can relate to function right away that if it has blue colors, it has partial positive charge characters. And if it has cyclic ring, like in phenylalanine, tyrosine, and tryptophan, these ones will be aromatic group. It's allowed for pi-pi or pi electron stacking. Like in เหมือนยางรถเนี่ยค่ะ basic car tire that can stack together up to the two rings together. So it stack on the top of one another into the pi-pi stacking. And this proline introduces kink in the helix. So it rolling back to the main chain. And when there is proline in the alpha helix, the helix will be bent because it rigid torsions of the main chain. So in comparison to glycine, glycine will allow flexibility, but proline will introduce tension or rigidity in the helix. And histidine, this one has two protonation states. So the PDB file format, normally it has the header, and then it will show you the compound class, which shows the molecule of the protein. Like in this one, protein is cyclic AMP dependent protein kinase. And it shows you what they are inside. Like this one has peptide inhibitor, and also it has fragments of inhibitory domain. It shows that in the PDB file header, you will see the organisms like this one, Bos taurus is in cow, and from the heart organ and expression system is Escherichia coli. So this is the expression system when the protein was cultured and crystallized. And there will be the literature reference who created these structures. And when you look inside the protein data bank file format, you can see that the beginning of the drawing would start at atom record. So the atom record will have leading of the line as atom, and it will have the atom ID and atom name, residue number, protein chain, and X, Y, Z coordinates. So this one just show that residue number 86 is glutamate chain E. And these are several atoms that I chose in amino acid side chains. And these atoms has it X, Y, and Z coordinate in Cartesian format. So it used atomic location in each X, Y, Z coordinate numbers in order to draw how the structure would look like in molecular visualization tools. So for glutamate, you can see that if it has the chemical sketching, it look like this, has glutamate and look like that. And histidine 87, histidine has 5 members. So this one has 2 protonation states in blue. So this one, histidine 87, glutamate 86. And if you draw it in several styles, this glutamate 86 and histidine 87 can be drawn as line representation, ball and sticks, and CPK space fill. So this ball and stick just basically drawn the atom with larger radii. So if the radii is equal, so for the cylinder and for the sphere, if it has equal radii, it will be called the stick representation used a lot in structural biology. And if you want to represent the true active site shape, normally they use CPK spacefill or van der Waals radii styles that basically have the atomic radii in proportion to the van der Waals radii. And when you look at it from the chemical perspective, you can see that people see it in bonds and in sticks characters. But if you are crystallographers and you want to show just roughly how the shape of the protein look like, you just omit all the side chains and looking at the main chain alone like this. So this is alpha helix. So you discard all the side chains. That would become the protein representation. This one is C alpha trace, basically traces of alpha carbon main chain. So this one, if they sketch roughly, you can see that you can see how many units there are, how many chains there are very quickly. Like this one, you can see the impression of 6 colors right away. And if you want to look at the secondary structure, such as the alpha helix in red, the beta sheet in cyan, you use ribbon or cartoon representation. Schematic has directions of the alpha helices and beta sheets. And van der Waals surface will be the van der Waals radii that sometimes show in coupled with the electrostatic properties. So you can see the partial negative charge or partial positive charge in colors. So let's zoom in. This is the alpha trace colored by chain. So you have the alpha helix like this. And so this is the unit like this, and you omit all the side chains. This is the C alpha trace style. And if you use the solid ribbon, you can see the helix like this and omit all the side chains you can see here, and the strand. So the strand, if the main chain go up to the top and going down, this is the beta pleated sheet. For the schematic, it link all the alpha helices into the rockets, has direction from N terminus to C terminus, and in arrows as in beta pleated sheet from N terminus to C terminus. So the van der Waals surface, this one have these characters. This one is colored by atom, oxygen in red, carbon in gray, nitrogen in blue. But if you use positive in blue colors, like this one, and then use negative charge in red, positive in blue, negative in red, like this, it will become the color by pKa, for example, which you can relate it to charge when you design the molecules to catch with the clef. So that's the basis of how to design the drug molecule to bind with the protein pocket based on the lock and key concepts. So example, this is how the structural biologists will, when you say you want to see the virus. So the virus comprises several protein and several nucleic acids and form together into a globular shape. A more simple representation, like this one, the matrix protein have structures in here, and this is nucleocapsids and have RNA structure inside. Let me play a video. Okay. So this is the animation created by Janet Iwasa. This is the HIV in comparison with the size of the T cells. So that representation of HIV, when it infects the T cells, it will go on the surface and it roll around. And then once the receptor binds to the T cells, and it finds the location, it will stop here, and then it will inject the red helix inside. So it recruits several proteins in order to inject the helix inside. Once it punch a hole, inject the genetic materials inside the T cells, and then the T cells will become infected. So it fuses the two membranes together. And then it gets in the T cells. So for example, if you are able to design the molecule that replace with this blue molecule, so you design a peptidic ligand in green, and it just wrap around the red helix, but it doesn't have the function in the same way as the blue one does. So by this way, you may be able to prevent the HIV from infecting the T cells because of the peptidic ligand just wrap around and doesn't allow the two membranes to fuse. So this one is coronavirus India variants video that I created. So let's see the protein structures that I shows with the spike protein. Tanramluk at Mahidol University. I'm reporting my mapping of the West Bengal B1618 mutations on the spike protein structure of coronavirus India variant. So the past 60 days, you can see that B1618 occurrence is here. And B1618 have three important mutations, which are deletion at 145, 146, E484K, and D614G. So what they mean? Coronavirus got the word corona from crown. Crown is this red part, which is called the spike protein. This spike protein can be understood from the structural perspective. You can see the red part here, which is the spike protein. If we flip it up, we can see the receptor binding domain in gray. And the South African UK variant has mutations in yellow, and the California variant has mutation in cyan. If the two combine at E484Q plus L452R, it becomes the B1617. And if the combination of E484K plus D614G in pink and deletion at the two green location, it will become the triple mutation West Bengal variant. So you can see that the structure is very beautiful and the beauty is extraordinary. However, it may mean hidden, scary consequences. So you can admire the beauty a little more time. The consequence of spike protein in B1618 lineage comprises the deletion and E484K, which are found to have immune escape variant, and D614G, which increases infectivity. So when we create the picture, we can look at the lineage report like this. And you can see that it has the structure. The first time Delta variant is called B1617.2. So this is the Delta variant. And in comparison with the Wuhan, the spike protein has these mutations. So I created those mutations based on this map using the structure of the spike protein S and mutate each location into the protein structure and use different colors for representation. So in this lecture, I will teach you how to make the animation of spike protein. So you can see the spike protein and you can also use the mutations and create the animation like I did early on. But that's after you install the PyMo on your machine. So I will teach you how to create the SARS-CoV-2 spike protein animations in the later on in the class. But I hope by this time, all of you are able to install the Discovery Studio and also the PyMo. So this is the mutations map that I showed you in the video. So I did it like in the animation, I did it in April. So that time it was still called the variants of concern. So the B1617.2 now is almost superset by the Omicron variant, but Omicron have more than 30 mutations. So it would be too hard a homework for you. I will just give you the B1617.2 homework so that it's easier for you to understand. But the mutation is already obsolete, like too old to be useful at this step. So let's say here, what you can look at each row is the various coronavirus mutation, various mutant, what we call various variant, like African variant, UK variant, and this is the location of mutation, like E484Q. So I taught you in amino acid side chains, glutamate or glutamic acid has the character E, for example, and N501Y, so N is asparagine. So you may not recognize it, but I have this little letter here, see, asparagine N, glutamic acid E. So in other classes, you should learn about these characters already. So it exists in the sequence report. So E484K is glutamate, and then K is lysine. So E484K that they report a lot is glutamate at position 484 mutate to lysine. So this will be your homework that you need to create and send me the assignment. So I will not give you the exam. I just give you the assignments and please do it correspondingly. So this one is called the structural model. So this is the spike protein model. And there is various physical models that if you meet me in person, I will teach you how to fold the protein by your hand. But since we are lack of the equipment at your home, so I will just give you an impression that there is a lot of physical model available that you can use the braces, you can use the laser to carve the structure inside. So these are physical models. So in the first invention of the structural models, this is John Kendrew and his structure of myoglobin. He used the rod and then put the beads on the rods like that and put atomic location based on the rod, because that time there is no computer that powerful enough to draw the structure. So that's always what it's like with the first invention. If you are the first one in the world, you have to invent the way to represent your idea, and it's always hard. But after we have a lot of computer program, these ones become easier. So this one example of cytochrome B562. So we have the structure start from N terminus to C-terminus. This is the representation of colors called rainbow representation. You go from ม่วงครามน้ำเงินเขียวเหลืองแสดแดง so แดง and go from red, green, yellow to blue. So start from N, green, yellow to blue. So this one's color representation also available in the PIMO. So this, you omit the side chains and you just show the main chain alpha carbons. So let's go to RCSB protein data bank. And then on the right-hand side, you can download file, PDB file. And you can also see the molecule in JSMol. So I cannot see at your computer, but you can visit the protein data bank rcsb.org now. And in the search panel, put 256B. This is the example of cytochrome B 256 complex. And you can use JSMol to visualize the structures. And normally when we meet in phase, that's the structure that I will taught you using the decorative ribbons to make the physical models with this 256B structure. I will show you the example of what they are with PP homework. So you go to courses, go to assignment gallery in ajanvi.wordpress. You can see that PP have made a lot of structure, model, and animations. So this is cytochrome 256B that Run P made in class. So you use decorative ribbon, use the pen, and then you fold the ribbon around. There is several other structures like this one, integral membrane protein, this one. So they are all your Run P, Rahat 62. So there's also other assignment, this one. So I also taught how to make the CRISPR-Cas9 animation and poster. So this is the example of your homework. So this is an animation. They are all your Run P. So you can look at the previous assignment of PP from here. There's problem with my graphic card. Now, let's come to the derived database. So the derived database that I will teach you is called CAS, stands for Class Architecture Topology Homologous Superfamily. They classify these shapes of protein into mainly alpha helix, mainly beta sheets, alpha beta sheets complex, and few secondary structures. So if you go to CAS database, casdb.info, you can see that there is the class 1 mainly alpha, class 2 mainly beta, class 3 mainly alpha beta. And if you expand the button of class 3, you can see that there is the, what we call the domain. So you can see row, super row, sandwich, prism, boxes, horseshoes, propeller. So this is the shape according to the protein structure fold. So if you want to create the physical model by yourself, you can sift through this CAS database and choose the shape that you want, and you fold the protein by hand. And then you can have the structure like PP does, but we don't have ribbons here with you at home. So I will just show you that it's doable by going to the CAS database. So this is the secondary database. So protein data bank is deposited by the crystallographers. but the CAS database is deposited by the one who process all those data and classes into the different folds. Now, practical one, searching when the sequence is known. So the example I will teach here is sickle cell anemia. Sickle cell shape or blood, red blood cell look here. Red blood cell have the sickle shape like this. This red blood cell normally will have the donut shape like this, but when it becomes the sickle shape, it will be stiff and sticky and tend to form clumps. It leads to blocked blood vessels and cause pain, serious infections, and organ damage due to the sickle shape red blood cell clump together and clog the blood vessels. So the structural basis of sickle cell anemia. So this is the hemoglobin beta as in packed together, and it's from fibrous. So why is it called fibrous? You go to protein knowledge base, uniprot.org, and search HBB_human. So let's open your web browser now and go to worldwideweb.uniprot.org and then search HBB stand for hemoglobin beta_human. So if you search this HBB_human in Uniprot KB and you scroll down to around the 2/3 of the page, you can see the sequence and you can click download faster. So after you download faster, you can see these sequences. And the hemoglobin sickle cell shape is the hemoglobin beta that have the first E of the peak mutate from first E to V. So if you use your keyboard, get this sequence from the FASTA and mutate from E to V, you can search how many hemoglobin structures that causes sickle cell anemia are deposited in the PDB. And you can also do the homework. What is the part of the protein that these amino acid mutations from E to V occurs? And the assignment that measure how well you understand the lecture and how you can apply the knowledge into further understanding of structural perspective is this question. Discuss why normal hemoglobin glutamate 7-aspartate 73 is less likely to form fibers than hemoglobin C Harlem. So I will teach you how to do this by Discovery Studio. So you just go to this PDB sum at EBI. So you search PDB sum from Google and copy this sequence and put inside the search by sequence panel. I will show you how. We go to Uniprot and then go to Uniprot KB and search HBB_human. By searching Uniprot, you can scroll down and this about 1/3, you can download the sequence in FASTA like this. And then you copy this section. So this is the header of the FASTA, describe what the sequence are, and this is the amino acid sequence. You copy and paste in the notepad. And then remove all the white space and enter line character. And then mutate the first E to V with your keyboard. So you can use this sequence after mutation as your query to search PDB sum. In PDB sum, you can have this search by sequence. So when you do your homework, you search by sequence here and click search. So this is the sequence of hemoglobin sickle cell with mutation from E to V. So by only 1 point mutation from E to V, the patients will change from the normal person into the one very sick with organ failure just by 1 amino acid mutations that cause aberrant hemoglobin beta. So the way to understand this panel, you want to search for sequence with 100% identity with the hemoglobin sickle cell. So that is this structure, 1, 2, 3, 4, 5, 6, 7, 8. So we want the structure with only heme and nothing else. So we want to HBS be this one, to HBS, high resolution crystal structure of deoxyhemoglobin S. So you search to for HBS like this. So once you click to HBS, you can download by this link. So you click on link and PDBE like this, or RCSB, doesn't matter. So that would allow you to download the structure. So you click download here, PDB file. And then by open this, you can show in folder and then open with Discovery Studio. This is the panel of Discovery Studio. You go to view, hierarchy. So this hierarchy will have this panel view hierarchy, see? And there is series of protein chain. So this is the unit cell with 2 HBS structure, chain A, chain B, chain C, chain D, chain E, chain F, chain G, chain H. When you do your homework, this one, aspartate 73 asparagine chain B. So you go to chain B and go to 73 first. So let's view display style and then off for this one first. And then go to 73. Go to 73. Aspartate mutate to asparagine. So view display style and then bold and let's say CPK and then color pink. Okay. Okay. So now you can see that this pink amino acid is here. So this is aspartate 73 asparagine. The next one is E7V chain H. So you go to chain H and go to E7V. Chain H, the first chain H in the tab. E7 คือ glutamate 7 and change to valine. So this one, if you have methionine as the first, the number will be shift from E7 to valine 6 because it has glutamate as the first number. So valine would be the V here. The first E mutated to V, so it's valine 6. And if you click here, you change by view, display style, color, cyan, click OK, and then CPK, apply. Okay. So this is the homework that you do with Discovery Studio. If you right click and you drag by right click, you rotate, you can see the reason why this you can see the reason why the D73 and E7V chain H once mutate, the normal hemoglobin doesn't form fiber, but this one tends to form fiber. Because the characters of normal hemoglobin, if these ones become negative and negative, the same charge will push one another, and so they swim in the blood better. But once it becomes hydrophobic and uncharged, it will tend to attach together and form fibers. So this is the homework that you need to send me as assignment in order to get the assignment done for question 1. So far, is there any question? So far you go to UniProt, and UniProt at the 1/3 of the page, you can see FASTA, sequence FASTA. โอ้มันบลิงก์มันเด้งเนี่ยนะคะเดี๋ยวนะ1/3 ของเพจนะคะจะมี fast 1/3 of the page has FASTA. Someone open the microphone. So you can see FASTA at 1/3 of HBB underscore human. You click on FASTA. Now you can have this sequence with new line characters. So you want to remove all the new lines and mutate the first E from peak here. E7th, you want to mutate the first E to V. So you copy it, and open Notepad, and then Ctrl V, remove all the new lines, and then mutate the first E to V. And then you copy, and then go to PDB sum. In PDB sum, that is the search by sequence. Ctrl V, and then search. Once you search, the ultimate goal is to find PDB code. This PDB code, you can Google it. You don't need to come here. You can to HBS and Google it. But the key is you need to know what PDB code that has the right content that you look for. So this one we search by sequence of sickle cell sequence, and then the 100% match means this one has exactly the same sequence that you put in. So exactly the same sequence of this page. Every letter is 100% match.
อาจารย์คะตรงจุดนี้ขอถามนิดนึงค่ะไอตรงเปอร์เซ็นต์ identity อะค่ะมันจะมีลำดับที่11ที่เป็น100%เหมือนกันอันนั้นคือทำไมไม่เลือกนะคะ
มัน100%แต่มัน145อะมิโนแอซิด
อ๋อคือเราต้องการ146เพราะว่าเราต้องการมิวเทตแค่ตัวเดียวใช่ไหมคะ
คือเราต้องการทั้งเส้นยาว146 the total length is 146 letters so this is 100% match but one letter missing
อ๋อขอบคุณค่ะ
so now we want to HBS right so you can click here and go by several steps or you can Google to hbs.pdb so you can to hbs.pdb and Google you can find right away but you need to know first when you want to search by sequence how to do it so you go to HBS like this and then you go link you can link out or you can Google purely to HBS you can get to HBS right away when you go PDBE so you download PDB file and you open once you open you can see this you click view hierarchy and then you can see this panel and what you want is chain B number 73 and view display style and then CPK ค่ะ
ดาวน์โหลดยังไงนะครับ
ยังไงนะคะ
ดาวน์โหลด PDB ไฟล์แล้วยังไงนะครับ
อ๋อได้แล้วก็ Open ออกมาเลยค่ะคือดับเบิลคลิกเข้าไปที่เครื่องมันก็เปิดเลยค่ะหรือว่าเราคลิก Show in folder ก่อนแล้วก็คลิกขวาก็ Open with and BioVia Discovery Studio คลิก OK
ของผมยังไม่ขึ้น Biovia เลยครับinstall Discovery Studio แล้วนะ
ใช่ Discovery Studio ไม่ต้อง BioVia ก็ได้มันคือ Discovery Studio แต่หลายเวอร์ชันชื่อไม่เหมือนกันคือเปิดด้วย Discovery Studio ค่ะเปิดได้ไหมคะ
สักครู่นะครับ
ก็ when you open it for the first time it will ask to test your graphic card or something like that just click yes okay yes okay ไปให้หมดค่ะแล้วก็ you can open this one so you go to chain B and then click on residue number 73 click on the left hand click it will be select on the right hand click it will be rotate so right hand and drag it will be rotation and it's just like playing games so are you up to this point yes number 73 view display style and then click on atom tab click CPK and then custom and click your colors any colors click OK and then you can see the location of these amino acid side chains and then you can do your homework number 1 so chain B and then the first set of chain H you want to go to chain H glutamate 7 mutate to V so you to go to glutamate number 7 which is mutate to V and view display style. So the reason why you use valine 6 is your input sequence has the first E mutate to valine. And this valine is number 6 because it has methionine here as number 1. So valine 6 is basically E7 in that numbering. So you click View Display Style, Custom, Blue, and click CPK, click OK. See? And when you describe in the homework, it's because the charge characters of these two in normal hemoglobin, they tend to push one another because of their charge. So normal human, these two doesn't form fibrous. In case you want me to look at your screen, share screen, multiple participants. So you all can share your screen with me, and then I can help you with your visualization panels.
อาจารย์คะเมื่อกี้ไม่ทันค่ะไปที่บอลแอนด์สติ๊กหรือ CPK นะคะ
CPK ค่ะ Ball and Stick มันจะเล็กกว่า CPK มันจะเป็น Wonder Wow หมดเลยมันจะเป็นลูกกลมๆใหญ่ๆติดกันแต่ถ้า Ball and Stick มันเห็นไม่ชัดแต่หนูเลือกมาได้ถ้าหนูเข้าใจว่าหนูทำอะไรลงไปเลือก CPK ก็เห็น Ball and Stick ก็จะเห็นชัดน้อยกว่าแต่ก็จะเห็นเหมือนกันค่ะนี่นะคะshare screen number 2 share. So in Discovery Studio, you go to display style. These ones, if you want to change the protein, you go to protein tab. If you want to change just the atom, you go to atom tab and choose CPK. So that's just atom. But before using that, you need to select what you want to change first before you view display style. If you click A and click Shift and click at the end, let's say H, you can select all. So you can click Shift, you can select in tab. If you click Control, you can jump from 1 to A to D to E, for example. You can click Control, you just choose some options, but you click Shift, you choose from the first click to the last click. So Shift, choose all. Control, choose only selected ones. Now, you click to MBS all. You can view display style, and then in the protein tab, you can choose schematic and see what it does. And right click on the background, left click on the background one time. Once you left click, you deselect it. So this is schematic. You can see rockets. And you can select all again. So the first set is protein. The second set is ligand. The third set from A to H is water. So the crystal is crystallized in water. The third set is water. So A to H in the first set is protein. A to H from the second set is the ligand, heme, see? The A to H in the last set is water. So if you select the protein and you view display style, you can also view it by color by rainbow. and then show solid ribbon, rainbow. Click OK. And click on the background one time. So you can see color by rainbow. You can click view, spin. Now let's we'll spin stop, and then so far, do you need any further help so that we can have time to do spike protein animation? No? So for batch downloading, so what if you have the set of proteins that you want to download? You want to batch download your protein instead of only one structures. So that's come the concept of ontology. Ontology is a controlled vocabulary that explicit and carefully defines sets of terms to distribute queries among several databases. So if you go to Quick Go, search EBI Quick Go, and you search GO colon 0004672, you can see the protein kinase activity like this, GO 0004672, and you can see the map of function representation. So gene ontology is classified into 3 classes that are molecular function, biological process, and subcellular locations. So each term is a relationship from the bottom up to the top. For example, if you choose GO 0004672, which is a protein kinase activity, it will go up T levels up to the top, which is molecular functions. So protein kinase is a kinase activity or phosphotransferase activity, and it containing phosphorus groups is a part of the family that has transferase activity. Transferase is a class of catalytic activity compounds, and catalytic activity is a part of molecular function. So if you know the GO terms, you can download anything with have that term. And each PDB code has much more than one GO terms that associate with it. So it will be the categories that you can batch download based on the GO IDs. So if you use GO 0004672, you can have like 50,000 structures, for example, that have protein kinase activity, and you download the whole class of it. So that's how you download the set of protein. So example, you go to RCSB protein data bank, and then search browse by annotation, and then click on molecular function. Under molecular function, you can put 0004672, and then it will expand the protein kinase activity panel in the bottom. And then under cyclin dependence kinase activity I will ask you how many entries are there that are cyclin dependence kinase activity with less than 1.5 angstrom resolution. The resolution is the quality of the structure. The smaller number is the best. So less than 1.5 angstrom is the ability to define the 2 points that are 1.5 angstrom apart. So the one with less than 1.5 is the super resolution structure. So this is the very high quality structures. I will guide you through by clicking in the following lecture. So you click download and then you can get the whole set of 0004672, for example. Okay, let's go to RCSB. So, now, can you see RCSB? Search, browse, annotations. And then you go to molecular function. And then you click gene ontology ID 0004672 and click here. And then it will expand this one. You click on the triangle pin and go to cyclin dependence protein kinase activity. And then you click here. Now, I ask you less than 1.5 angstrom. How many structures? And then you answer in the homework. And here, these are all the structures with cyclin dependent protein kinase function. So if you go by gene ontology, you have exactly the function that you are looking for. If you go to Google, you can hit into hundreds of hits without knowing which one is the right one. But if you go by gene ontology, you cannot miss it. And now, I will show you how to do ligand interactions. Let's say, click play here. So this means 0.5 to 1 to 1.5. You click this 2 and then you click play. So it means you want to select only these 2 sets. You can also set based on organisms, sets on SCOP classification. So that's another way of CATS4. So CAT and SCOP search the protein by the shapes. So this one, if you go to the structure, let's pick 4FKL and click download file. เราต้องการที่เป็นคลิกดาวน์โหลดมันเป็น GZ เนาะ we want to have the 4FKL and download only the PDB format. So you search PDB format and then click open. Now you can see this is the protein with the ligand. And if you go to receptor ligand interaction and click ligand interaction. Click OK. You can see the ligand interactions. But this one, I will not ask you in the homework. So I will teach you how to do this, but you don't need to capture or do anything with this. So basically, these ones will show you what molecular interactions they are. Like this one, the hydrogen bond, there is pi-pi stacking. You can see the hot pink, pi-pi stacking, phenylalanine and this ring stacking together. And hydrophobic conventional hydrogen bond. So you can see in color code. So this one is the ligand interaction between the drug and the protein. So the drug is the key, protein is the lock. If it fits in lock and key concept, it may inhibit the protein by the structure. Let's go back to our slides. So question number 2, how many CDK kinase with resolution less than 1.5 angstrom are deposited in the PDB? About heterogens. So heterogens or the het records of the PDB file is the non-standard residues such as prostatic groups, inhibitors, solvents, or ions. Is not one of the nucleic acids, not one of the standard amino acids, and identified by 3-letter code in het atom column of the PDB file. So heterogens can be searched in the web server that I create. So in the research highlight, we will show that there will be one group that shows the server called Manora. I will demo a little bit on Manora, and then we will have time for spike protein. So, so far, is there any question before we continue with the structural exploration of SARS-CoV-2 spike protein using PIMO? Do you want to have a break?
Yes อาจารย์
ค่ะ งั้นเราเบรกสัก10 นาทีนะคะ
อาจารย์คะรบกวนอย่างหนึ่งได้ไหมคะ อยากได้สไลด์แบบเป็น PDF อะค่ะ คืออยากจดลงไปด้วยอะค่ะ
ได้สิ คือตอนนี้สไลด์อ่ะมันจะเป็น
มันจะเหมือนเป็นแอนิเมชันให้ดูไปเรื่อยๆใช่ไหมคะ
ใช่ ก็คือสไลด์ตอนนี้เป็นแอนิเมชันค่ะ หนูอยากได้แบบเป็น PDF แล้วเขียนลงไปแล้วหนูจะ
อยากได้เขียนด้วยอะค่ะ
อ๋อ
เพราะบางทีที่อาจารย์พูดมารู้สึกว่าอยากเทคโน้ตลงไปอะค่ะ
ได้ค่ะ ก็เดี๋ยวจะมีวิดีโอที่อาจารย์จะ release ออกไป แล้วตรงนี้สไลด์ก็คือจะเป็น exactly the same ที่อาจารย์พูดก็คือจะมีทุกอย่างเลย เดี๋ยวอาจารย์ save เป็น PDF ออกไปแล้วก็ deposit ไว้ใน Google Class นะคะ ซึ่งข้างหลังเนี่ยค่ะก็จะเป็นเดโมหมดเลยแล้วทุกอย่างที่อาจารย์ทำถ้าเกิดว่าหนูตามช่องแดงๆอย่างเงี้ยค่ะหนูจะทำได้หมดเลยด้วยตัวเองโดยที่ไม่ต้องมีอาจารย์อยู่เลยค่ะ
ได้ค่ะขอบคุณมากเลยค่ะอาจารย์
ค่ะแล้วหนูจะปริ้นทันเหรอคะ
เดี๋ยวหนูเปิดใน iPad เอาค่ะ
อ๋อได้
ดูไปด้วยค่ะ
ได้ค่ะ
ขอบคุณค่ะ
โอเคค่ะ so now the structural exploration of SARS-CoV-2 spike protein we want to help you to visualize and explore the structure of SARS-CoV-2 spike protein. So this is the coronavirus as I showed in the previous videos. And it consists of spike glycoprotein as the triangle here. Other parts we will not focus in this lecture. So Manora showed you the main protease, but this demo, this practice would be on spike protein. So it's the crown here, the pink one. We will download the structure of spike protein, and then we will use PyMo to visualize it. So PyMo is written by Warren Delano, but he passed away quite a while ago, and it's continued by the Schrödinger company to write and maintain the PyMo platform. If you want the latest version, you can download it at pymo.org, but it requires license, and each time you have to continue like half a year to enter the license and download the license every half a year. So you can download on Windows, Mac, and Linux, and you download the license file, you put in the forms, and then this is the educational version that I distributed to you. So you open the file with PyMo, and then you search the PDB code. So this one, the main protease has the heart shape, but I will focus on the structure here, 6ZP2. Can you see? 6ZP2. So you Google 6ZP2.pdb in Google, you can see this structure. You can Google now, and let's open it on PyMo and play it together. So Google 6ZP2. So you Google it. There's hundreds of them, but I use this one for illustration. So you show in folder and then open with PyMo. So PyMo will have two panels. The graphical representation panels and this command panel. So when you do the structure, it will have action A, S show, H hide, L label, C colors, and then you have sequence here. In the sequence, you scroll it on the right-hand side. So this is chain A. From chain A, you go along here, and then it will start again with chain B. And you go until it's end at 900 something, and then you start again with chain C. And if you want to select certain atoms, certain residues, you just click and drag again to deselect it. So this is how you select the sequence by without using the command line. So you just scroll it for a rough idea. So now I want to use this 6SP2 and hide everything and just shows cartoon. So I want to hide all atoms and shows just cartoon. And then you can click on 6sp2 line again and click colors and choose the colors that you want. You can right click and drag, that is zoom, left click and rotate, left click and drag, that is rotate. So this is how I create the molecular visualization of spike protein. So let's go to our slides and see again which position we want. So this one, action, show, hide, label, color. And if you click on the left-hand side and rotate, it's rotate the structures. If you right-click and zoom, it will show it's in larger and smaller structures. So the structures that we want to play with is this B1618, which has deletions at 145, 146. So we omit this one, we just use E484K and D614G. So what they means, we want to go to chain A at position 484K, E from red negative to K in blue, or in whatever colors you want, just show the position where they are. And this 614G, we want to show the 614 mutate to G. So going back to our structure, we want 484 and 614. So we go to this panel, go to chain A, E484K, and then click show sphere. Click color, add selection, add selection, color, green, sharp green. So this is E484K, and that is 3-degree rotation. So 3-4 rotation means that is 484 occurs 3 times. We go to 484 again. E484K, and then color, green, and then show sphere. And then we go another one, 484, and show sphere, color, green, like this. So you can see the location of E484K, which is a very important location for spike protein characters of the one that are very have high infectivity. And you need to do another fold for D614G. And then the way to do animations is go by open this one, click scene, append, and then you rotate it a bit to the proper position and scene, append, and then you go up to another view, scene, append, and then go to append, program, scene loop, notate, 630 degrees, and then click play. So it go from top view and front view that you append, and then it go to the bottom that you append. And then it show the bottom again. So with the latest version of PyMo, I hope you can save in MOV or in MP4, but I wasn't sure because it wasn't work before. But you can screen cap, like use screen capture tool to crop this, and you can send me the video. So this is all the homework. The first one is hemoglobin sickle cell, 3 questions. And then the second one, how many CDK4 with less than 1.5 angstrom resolutions? And this one. So this one's, you can capture it and put it as poster or just animation alone. I don't mind. You just try to make the spike protein. It doesn't important that you need to do 618, B1618. You can attempt other spike protein, but the Omicron one is like 30 position and 3 degrees. So it's 90 residues. So that's too much. So you just do the simple one and then try to do the animation like this. So this is all. Let's make animation. And then in the rest of the class, I will assist you. You can share your screen, and then I can assist you how to make the animation. And in summary, you have learned about the RCSB UniProt, CATH, PDBE, and PDBsum. So this is primary data source. This is sequence database, the full database called CATH, PDBE that you can use to download the PDB file, and this one PDBsum that you can search by sequence. And you already know how to find the structures if you know the sequence, function, fold, and the ligand. And you learn about the importance of structures to diseases, drug side effects, active research in the field, and you know how to use molecular viewers, PyMo, to make covers and graphics illustration. Okay, so now I think we have about 45 minutes for doing all the homework together, and I will help you as much as I can. So you just share your screen in case you need help. Otherwise, everybody deserve the time off tomorrow ตรงนี้นะคะ แล้วเราก็เปิดหรือเราจะเปิด PyMo แล้วเรา open file ก็ได้ พอเราเปิดขึ้นมาปุ๊บพอเราเปิดขึ้นมานะคะตรงเนี้ยฟังก์ชันที่เราจะดูอ่ะมีแค่4ปุ่มตรงนี้ก็คือ action มันก็จะเป็นพวกแบบ zoom orient อะไรพวกนี้นะคะก็ไม่ต้องไปสนใจมากอันที่เราสนใจคือเราจะ high lines ไปก่อนก็คืออันนี้เป็น lines representation เราสามารถเล่นได้หลายอย่างตรงนี้มันก็เหมือน Discovery Studio แต่ถ้ารู้2ตัวต่อไปก็จะทำได้ทุกตัวเลยก็คือตรงนี้ high lines แล้วเราก็ shows สมมุติเรา show cartoon พอโชว์การ์ตูนเนี่ยค่ะมันก็จะเป็น ribbon representation พอเรา right click เราก็จะซูมออกมาได้เนี่ยค่ะแต่ว่าถ้าเราซูมออกแล้วก็ right click แล้วเราก็ถอยลงมาเห็นไหมลองเล่นไปด้วยกันแล้วเราก็หมุนได้ทำอะไรได้ทุกอย่างทีนี้สิ่งที่เราอยากทำก็คือพอเราได้เป็นการ์ตูน representation of the main chain แล้วเนี่ยสิ่งที่เราอยากได้คือเราอยาก color ใช่ไหมคะเราก็อยาก color ให้เป็นสีสมมุติสีเทาไว้ก่อนพอได้สีเทาปุ๊บหนูก็จะใกล้วิดีโอของอาจารย์แล้วคือเราก็ไม่สนใจอะไรเสร็จแล้วเราคลิก sequence ข้างล่างนี้นะคะใน sequence เนี่ยก็จะบอกเหมือนกับ view hierarchy ของ Discovery Studio แต่อันนี้จะใช้ยากกว่า on the top there would be the sequence from PDB structure to ZP2 and chain A and then you want E484K so you scroll to 484 and find position of E amino acids and then you click show sphere and click colors and then you choose pink so you have this position of E484K and each time you append means you want that scene to be the point of camera capture so for example if I want to show only this one I will rotate to the top first like this and then I will use the right hand click to zoom until I want the size that I want in right hand click and then I will click scene append and then I will rotate until it's on the top view or on the front view and then I will click scene append and then another scene that I want maybe on the bottom I open until the bottom and then click scene append and if I want the pink one to show too I click on the pink that is select and right click and click orient and deselect the here and then click scene append and then you click movie program scene loop notate 30 degrees 2 seconds so by this one you click play it will stitch all the scene that you append into the videos so you go to the front and then you go to the bottom and then you orient at the top position and see this amino acid location. Okay, I will have this lecture in recorded videos. So I think you can follow every step very easily just by follow the screen recording. So is there any query that you want me to show you again? มันใหม่มากจริงๆต้องไปเจอกันนะ stop share screen 2 here and then I have post the handout and I also have posted the interactive slides. In interactive slides, you can click here and every step will be exactly the same as my slide. So you can click on the structure slide 94 and then you can see left click, drag here, click drag on the right hand zoom, left click rotate, and then I told you the 6ZP2 structure, and then I will have the steps of E484Q, L452R, and then I show how to choose the residues, right? And then make colors, and then click right click, click orient, right hand click on the position and click orient. And then you just append each scene. And then at the final, after append all the scene, you go to program, scene loop, notate 30 degrees over 4 seconds. And then it's end. So very easy. Anyway, you want to try, or you want any, you can share your screen and then I will show from your friend's screen, and then after you see like 3 of your friends, you probably be an expert by that time.
โอ้โหตัวเองกดอะไร มันเป็นแบบนี้ค่ะอาจารย์ อาจารย์เห็นที่แชร์ไหม
ยังรออยู่เน็ตช้านิดนึงเนาะสวยออกมันไม่ดีตรงไหน
คือหนูมันไม่เหมือนกับของอาจารย์นะคะอาจารย์ไม่ทันหนูก็เลยกดเล่นเอาเองค่ะ
ใช่คือ structure ที่หนูต้องการคือ XZP2 ไงคะก็คือเล่นไปเรื่อยๆอย่างนี้แหละค่ะก็ทำ animation มาหรือจะไม่ทำhow about you don't want spike protein but you want to do video of some other protein that you want to play with, you can do any other animation as well. I just want to help you to do the easy ones, but in case spike protein is too hard,อย่างกล้อง you can use มีใครเปิดไมค์ you can use the protein of your interest. So you have the hemoglobin assignment that shows your ability to understand the charge and shape and structures, and then a little capture on the number of structure of CDK2, and then this animation, that would be the end of my requirement for fulfilling the class objective. So in this one, you can click 6ZP2, and then you can, and then you hide 1 TIL by clicking again at 1 TIL. So if you want to hide, you click on 1 TIL again. Yes, and it's gone. And then you click 6ZP2, and then you right click center. Click on 6ZP2, add it, make it visible, mix center. Yes. มาไหมon the right hand side, and then actions, click hide lines, and then shows S ribbon or cartoon, yes. Then click color on the right hand side, and then choose colors ข้างล่างก็ได้ค่ะมันก็จะไปทั้งอัน if you choose the above one, it will show by secondary structures. Yes. and then you click on the right hand side on the bottom S
ตัว S
ตัว
อ๋อ
S
ตัว S โอเคขอบคุณค่ะ
and then 484ก็คือข้างบนมันก็จะขึ้นมาเห็นสีส้มนี้ป่ะแล้วก็
หนูเห็นแล้วค่ะอาจารย์อันนี้
เอา V ออกแล้วก็คลิกโชว์แล้วก็คลิกสเฟียร์ลด down นะสเฟียร์โอเคแล้วก็คลิก color แล้วก็ใส่สีค่ะใส่สีไหม
เห็นแล้วค่ะอาจารย์
แล้วก็จับให้มันเป็นรูปสามเหลี่ยมเป็นรูปสไปค์แล้วก็แอปเพนทีนึงแล้วก็คลิกไปคลิกมาแล้วก็แอปเพนอีกทีนึงแล้วลองเพลย์ดูก่อนหลังจากนั้นเราจะเพลย์ที่ advanced left click ไม่ต้องคลิกตรงนี้คลิกที่แบ็กกราวด์ออกไปข้างนอกเลยโอเคคลิกที่แบ็กกราวด์ทิ้งแล้วก็ซูมได้ทำอะไรได้ถ้าเราคลิกที่ตัวสีเขียวแล้วเราคลิก orient คลิก1 ทีแล้วก็คลิก orient มันก็จะซูมเข้ามาที่ตำแหน่งนั้นคลิกข้างบนก็ได้ข้างบนตรงตัว E ข้างบนก็ได้ตัว E ข้างบนสีเขียวแล้วก็คลิกขวา orient แล้วก็คลิกขวาแล้วก็ drag เพื่อซูมให้มันเล็กลงหน่อยไม่เอาอันนี้ออกคลิกที่แบ็คกราวด์สีดำ1 ทีเพื่อเอาอันนี้ออกไปแล้วก็คลิกขวาแล้วก็ drag ไม่คลิกขวาค้างเลย
คลิกขวาค้างใช่ไหมคะตรงนี้
แล้วก็ drag
เหมือนซูมออกเนี่ยนี่นี่ซูมเข้า
ก็แล้วก็คลิกแบ็คกราวด์1 ทีให้จุดมันหายไปก็ได้แล้วก็แอปเพนไว้อะไรเงี้ยค่ะก็คือพอมันมีมูฟวี่เราไม่อยากให้จุดนี้มันขึ้นมาด้วยมันไม่สวยไงแล้วก็คลิกที่แบ็คกราวด์1 ที
วิธี
ไปคลิกตรง E ไปคลิกตรง E ข้างบนค่ะคลิกซ้ำลงไปอีกที
มันหาย
ได้เหมือนกันแล้วก็ Scene Append ค่ะ Scene Append ไปที่ข้างบนตรง panel ประตูเขาเรียกอะไรหน้าต่างที่เป็นหลายคอมมานด์นะคะหน้าต่างที่เป็นคอมมานด์นะคะ
นี่ค่ะ
หน้าต่างสีขาวขึ้นมาหน้าต่างสีขาวที่เอาไว้เปิดไฟล์แล้วก็ Scene Append แล้วก็ถอยออกมาหน่อยจากตรงนี้นะคะก็คือตรงรูปอะค่ะตรงรูปสีดำอะค่ะเราไปให้มันเป็นสามเหลี่ยมสักทีหนึ่งไว้ให้มันรู้ว่าสตรัคเจอร์มันเป็นอะไรอย่างเงี้ยที่ขวาเซนเตอร์ก็ได้แล้วก็เริ่มใหม่ขวาเซนเตอร์ก็สครอลหน่อยไหมคือตอนนี้เราต้องการจะให้เห็นภาพเป็นสามเหลี่ยมอะค่ะลองสครอลนะหนูต้องสครอลตรงปุ่มกลางสครอลค่ะสครอลตรงปุ่มกลางเนาะเมาส์ปุ่มสครอล
ตรง
ใช่เมาส์ปุ่มกลางสครอลก็จะเป็นแบบนี้แล้วหนูต้องการให้มันเป็นรูปสามเหลี่ยมให้คนรู้ว่าเนี่ยมันคือโคโรนาไวรัสเนาะหนูก็จะต้องคลิกให้มันเป็นรูปสามเหลี่ยมแล้วก็ดูสามเหลี่ยมไม่ได้ดูแบบเบี้ยวอ่ะก็คลิกซ้ายขยับหน่อย
ซ้ายขยับ
คลิกซ้ายแล้วก็ค้างไว้แล้วก็ยึบขยับคลิกซ้ายค้างไว้แล้ว drag เลื่อนไปทางซ้ายตรงทิศ11 นาฬิกานิดนึงซ้ายหน่อยนิดนึง
เป็นสามเหลี่ยมอย่างนี้ใช่ไหมคะเป็นสามเหลี่ยม
คล้ายๆมิกกี้เมาส์เลยนะคะ
ตอนนี้หยุดแล้วก็
ใช่ตอนนี้หยุดแล้วค่ะอาจารย์ทุกคนขยับอีกไหมคะ
ไปที่หน้าจอตรงตะกี้แล้วก็ Scene Append หน้าจอสีขาวตะกี้ค่ะ C append แล้วก็ซูมขึ้นมาอีกหน่อยหรือว่าจะถอยไปด้านหน้าก็ได้แล้วก็ scene append แล้วก็จบละกันบ้านมีแค่นี้
ซูมขึ้นมาอีกหน่อยซูม
ทำยังไงก็ได้ให้มันแบบสวยๆแล้วก็ scene append ลองเล่นดูเกิดเล่นเราไม่ชอบก็เปิดใหม่แล้วก็ทำใหม่ว่าเราเซฟเป็นแล้วเราต่อจากตรงนี้ไปได้แล้วก็ไป movie
อยู่ไหนอยู่นี่
โปรแกรม camera scene loop ข้างล่างscene loop โอเคอันนั้นแหละคลิกเลยเดี๋ยวต้องรอ
มูไม่ได้ใช่ไหมคะ
รอไปถึงด้านขวาสุดเลยมันจะมีส้มสักตอนหนึ่งแล้วหนูก็แคปเฉพาะตอนนั้นมาคืออันเนี้ยหนูเคยแคปตอนที่มันเป็นซีนที่สีเขียวเอาไว้ก่อนแต่ว่าพอสีส้มมันก็จะเป็นอย่างนี้ไงก็คือหนูสามารถ combination between cartoon ribbon sticks all the representation and you can click on and off and change several scenes and it will calculate and stitches the pictures in between for you so you can capture the middle scene and you can play 5x speed in YouTube or something so it's faster but in general if you only capture 3 or 4 scenes it will be very fast okay I think you are okay so you just capture the two last figures so it should be all right
ผมโอเคแล้วครับผมเข้าใจแล้ว
เยี่ยม
อาจารย์คะหนูติดปัญหานิดนึงค่ะว่าแบบมันเป็นจุดเดี๋ยวแป๊บนึงนะคะเดี๋ยวลองโชว์ไอ้ตรงจุดอันเนี้ยค่ะมันไม่เป็นก้อนกลมๆอะค่ะมันมาเป็นจุด
หนูกลับไปเลือกอีกทีหนึ่งแล้วก็คลิกโชว์เลือกตรง E แล้วนี่ค่ะแล้วคลิกโชว์ตรง select ค่ะตรง select ด้านขวา S นะคะS ตรง S นะคะ
อันนี้ใช่ไหมคะ
ใช่แล้วก็คลิก spheres ค่ะ
sphere ขอบคุณค่ะ
คลิก color แล้วหนูก็ change colors นะคะแต่ว่าอันเนี้ยมันมีโชว์สติ๊กเยอะอะค่ะสติ๊กมันก็อินเทอร์เฟียร์กับความเป็นสามเหลี่ยมนิดหน่อยเนาะมันก็ดูไม่ค่อยสวยเท่าไหร่มันปนๆน่ะ
หนูต้องเอาตรงไหนออกคะ
คลิก high นะคะตรง6 ZP2
ค่ะ
stick high stick ใช่ค่ะ
แล้วก็ต้องเริ่มจากมุมไหนคะควรจะเริ่มจากมุม
ก็
หรือแล้วแต่เราเลยค่ะ
แล้วแต่เราเดี๋ยวโชว์วิดีโออาจารย์ให้ดูอีกทีไหมคือวิดีโออาจารย์ก็ทำประมาณนี้เดี๋ยวอาจารย์ทำให้ดูวิดีโอสไลด์ที่28แล้วก็คลิก play ในสไลด์ที่28ของวิดีโอที่อาจารย์ส่งไปใน Google Class หนูก็จะเห็นว่าที่อาจารย์ทำ
from the structural perspective you can see the red part here which is the spike protein if we flip it up we can see the receptor binding domain in gray and the South African UK variant has mutations in yellow and the California variant has mutation in cyan if the two combine at E484Q plus L452R, it becomes the B1617. And if the combination of E484K plus D614G in pink and deletion at the two green location, it will become the triple mutation West Bengal variant. So you can see that the structure is very beautiful and the beauty is extraordinary. However, it may mean hidden, scary consequences.
เนี่ยอันเนี้ยที่อาจารย์ทำอาจารย์ก็ไปที่เขียวออเรียนชมพูออเรียนเหลืองออเรียนแล้วก็จะมาข้างหน้าอย่างเงี้ยค่ะเก็ตปะคะ ก็ทำยังไงก็ได้ทำไปทำมาแล้วก็แบบพากย์เสียงไปด้วยหรืออะไรไปด้วยอะไรเงี้ยค่ะมันก็ได้หรือว่าทำวิดีโอไว้ก่อนแล้วก็พากย์เสียงได้อาจารย์ไม่ได้หวังว่าให้หนูพากย์เสียงได้คืออยากให้ทำภาพเคลื่อนไหวได้ก็แบบบรรลุวัตถุประสงค์อันสูงสุดของคลาสนี้แล้วค่ะ ทำก็คือทำข้อ1 hemoglobin ข้อ2กี่ structure ก็ข้อ3 animation ของ spike protein หรือโปรตีนที่หนูอยากได้แต่ว่าอันเนี้ยสอนไม่ทันเพราะว่าทุกปีจันจะมี6 ชั่วโมงอันนี้มันอัดสุดๆแล้วมันได้แค่นี้อันนี้สอนไม่ทันแล้วอาจารย์ได้โชว์ปะไม่ได้โชว์นี่ พูดอยู่คนเดียวจะโชว์ให้ดูนี่ก็คืออันนี้คือการบ้านที่แบบปีก่อนๆสอนไงก็คือทำ CRISPR-Cas9 structure แล้วก็ทำเป็นโปสเตอร์ก็คือมันมีทริคอยู่นิดเดียวว่าเราเปลี่ยน background ให้เป็นภาพ JPEG จาก Discovery Studio แล้วเราก็เอารูปเนี้ยแปะลงไปแค่นั้นเองซึ่งตัว structure หนูก็ทำได้อยู่แล้วก็เหมือนกับมันก็จะมี surface นอกนั้นหนูก็ทำได้ก็คือเลือกตำแหน่งต้นตำแหน่งท้ายแต่ว่าเดี๋ยวแชร์ก็อันนี้อาจารย์คิดว่ามัน request มากไปหน่อยการบ้านมันเยอะแล้วอะค่ะคือถ้าส่งแบบปีก่อนๆก็จะต้องมีเวลานิดนึงเนาะตรง class work ตรงอาจารย์จะพูดแป๊บหนึ่งว่าทำยังไงแต่ว่าข้อ2โปสเตอร์เนี่ยเปลี่ยนเป็นเหมือนกับกี่สตรัคเจอร์ก็พอไม่ต้องส่งมาเพราะว่าอันเนี้ยทำไม่ทันเนี่ยอันนี้ก็คือเวลาที่เราได้เปเปอร์นี้มาเราจะมี PDF เนี่ยสตรัคเจอร์เนี้ยเวลาที่เราได้ภาพแบบนี้มาก็คือมันมี 1,369 residue แล้วก็ใส่สีทั้งหมดเป็นสีเทาก่อนเสร็จแล้วเราก็ใส่สีช่วง180ถึง308เราก็จะได้ตรงนี้มาถึง718แล้วเห็นปะก็คือเรา shift แล้วเราก็เลือกด้วย Discovery Studio ว่าเรา arrange ตั้งแต่ residue 180ถึง308เป็นสีอะไรแล้วก็เลือกตั้งแต่718ถึง1099เป็นสีฟ้าอย่างเงี้ยค่ะแล้วเราก็ใส่เสร็จแล้วเราก็ใส่ surface แล้วเราก็เปลี่ยน background จาก transparent ให้เป็น background รูปที่เราอยากได้ save image as desktop เขียนว่า image ถูกไหมเสร็จแล้วในภาพนี้ค่ะวิธีทำโปสเตอร์ค่ะก็ view display style graphics image คลิกโอเคเนี่ยค่ะแล้วเราก็ export picture ออกมาไงก็คือเราได้ภาพ aurora ตรงตะกี้อะค่ะแล้วเราก็ได้รูปตรงนี้ แล้วเราก็เซฟเป็นแอนิเมชันออกมาแล้วก็ทำโปสเตอร์ได้นะคะแล้วเราก็ทำโปสเตอร์ CRISPR-Cas ได้ด้วยวิธีแบบนี้คือถ้าสอนอย่างเงี้ยหนูก็จะเข้าใจว่าหนูจะทำแอนิเมชันหนูจะทำภาพนิ่งหนูจะทำสไลด์ทำโปสเตอร์เวลาไปพรีเซนต์ก็ทำได้หมดเลยค่ะ